Okay, so welcome to the session. Officially, it's a session on Global Eye Genetics Consortium, and I'm delighted to co-chair the session with Professor Paul Baird and Professor S. Natarajan. It's an exciting program in the sense that this has a very modest beginning in this part of the world. And I was fortunate enough to work with Professor Natarajan and Dr. Takeshi Iwata from Japan, who is our co-conspirator in the whole effort. And then Professor Paul Baird has joined us shortly after. So at this point, many of you know Professor Calvin Pang. He's the first speaker. He's unfortunately he's not been able to join us due to all this coronavirus issue. Professor Pang is based in Hong Kong, and he's not. The delegation from Hong Kong, China, has not traveled. So we miss him here, but delighted to have the Vice President of Worldwide Global Eye Genetics Consortium, Professor Paul Baird. As many of you know, I'm the past president, and we have Professor Natarajan is the Secretary General. So all three of us here at the table have and continue to play a key role. So it is my great honor to introduce Professor Paul Baird from Melbourne, and a good friend, and we work very closely. And I'm going to leave it up to him to lead and start the session. Professor Baird.
Good morning, Paul and John. And I think I'm unfortunately placed in 3 halls, so I will be going in and out, and I would like you to excuse me. So, anyway, Paul, as John said, we had our what talk he's going to give, the Keratoconus International Consortium. We had the we had the meeting in Mumbai on February 10th. I hope Paul is going to talk about it.
Yes, thank you, Dr. Natarajan.
We'll represent you here.
For the invitation. Yeah. Aha, here we go. That's better. Okay, so what I want to do today is really talk about what you can do with collaborations around the world, and how this can be used for the betterment, I guess, of understanding a disease. So I'm going to talk about keratoconus. And some of you may have heard of this, some of you may not, but this is an example of something that we're doing with a corneal disease. So just to give you an idea, this is, if you see a side view of the cornea, it looks pretty normal, whereas once you move into more moderate advanced disease, you can see this nice sort of protrusion of the cornea sticking out. And I think anybody, and not as a non-ophthalmologist, could easily pick this up. It's not too difficult. And of course, as we go from normal vision, subclinical, with essentially a normal presentation of the cornea, through to mild keratoconus, through to advanced keratoconus, we can see more and more severity in terms of the disease pathology. And of course, a decrease in vision as well. Now, clinically, diagnosis is pretty much done based on a range of measures, either using slit lamp biomicroscopy, retinoscopy, keratometry, or topographic imaging, such as pentacams. And this is an example of pentacam imaging, showing a heat map of different elevations of the cornea across left and right eyes. So you can get a whole range of different maps. You can get a whole range of different parameters from these individual machines, and you can make a lot of use of that. Keratoconus is, even though it's relatively rare, it occurs in children and young adolescents. So it affects basically people for the rest of their lives, as opposed to aging diseases such as macular degeneration or glaucoma. And so the impact is basically lifelong. There seems to be an increase in male to female ratio of keratoconus, and there are ethnic differences around the world, and we don't really understand why there are ethnic differences, but certainly in Asia and the Middle East it seems to be more prevalent than in Europeans. And I think this goes with the ethos of GEGC in terms of trying to tease out different ethnic populations and what's going on in different ethnic populations. Prevalence seems to be increasing, and we're not sure whether this is because there is more keratoconus in the world or because there's actually better detection techniques, but it's been going up from 1 in 2,000 to about 1 in 30, some of the latest studies. And it accounts for the majority of corneal transplantations in Australia. In terms of non-genetic risk factors, you probably know if you're in the area that eye rubbing, allergy, connective tissue dysfunction and contact lens wear are important factors, but there's also genetic risk factors as well that have come from studies using twins, families, candidate gene studies, and also genome-wide association studies. And if we look at some gene associations that have been reported, you can see this great big long list here, but unfortunately most of these are not replicated and the results are fairly inconsistent, and so it really doesn't really give us a good handle on what the genetic cause of keratoconus is. So previous studies reported around the world are pretty much small, around about 100 patients, possibly 100 controls as well. They tend to be underpowered because of this size. Early moderate disease is not that well documented in many studies. Few studies actually combine multiple facets such as clinical, demographic, environmental or genetic risk factors. And so there's many unanswered questions in terms of how we define disease, how we actually look at subclinical disease, how we monitor progression, how we actually classify progression, and how we would actually use this information to improve clinical diagnosis and translation. So the KIC, or the Keratoconus International Consortium, was set up a few years ago by myself and colleagues, and really it was to invite groups all around the world to join together to form a consortium investigating keratoconus. And the idea was that we would collect uniform data fields so that we could talk between the different groups, which is very important. We would collect a defined set of clinical imaging risk factors, as well as genetic and bioinformatic biomarker data, if it was available. And this would lead potentially to new treatment options. And of course, we would like to translate these findings back to the patient. And we've already started doing some AI applications on the data that we've been collecting, our own data, but of course we can roll this out to this particular KIC consortium as well. So here's some little boxes in terms of a modular collection system that we set up, because we didn't expect everybody to collect all the data that we possibly wanted, so we defined this as a modular way. So everybody who comes into the study basically gets a standard eye exam in the clinic. They might also get a general health questionnaire. Now, that would be fairly standard information for a patient, but of course you can have other questionnaires that you can ask, such as quality of life, economic burden. You can obtain biometric measures if you wish, and you can also go further and start collecting biomaterials such as bloods and salivas if this is your interest. So the setup was really to ask and collect a standard data set, collect a defined set of core research fields. We asked all the potential investigators who are interested to come together and define what they actually wanted to get out of this particular consortium. We would collect retrospective and prospective data. The data remains a property of the individual members, so it's not something that I have control over. If Group A gives some data, then they actually still have control over that data, but they can share it if they wish with the other members. And the studies are basically driven by the members themselves. So the easiest way of collecting this data into a systematic way was really through this platform called Redcap, which is fairly standard now around the world. And it allows you to build questionnaires very quickly. It allows you to export data to common analysis packages. It allows you to schedule visits. It allows you to manage contact lists. And it's also mobile, and it's also web-based as well. So it's a very powerful technique. It's very simple to use, and anybody can enter the data. And here's an example of just page one, which is really just a basic details on an individual, such as their name, the hospital ID, email, etc., etc. So it's just a series of drop-down menus, if possible, to make life as easy as possible for the collecting person. And what we did was we actually collected a whole series of different modules that are listed here, so registration, medical history, and so on and so forth. And we also collected longitudinal data here across different visits. So this gives us an idea of what we need to collect. In terms of data that we've already collected, retrospective, we have 400 individuals from 2 sites in Australia and also Hong Kong, Calvin Pang. We also have some prospective data from China, Australia and Algeria, and we also have another 400 samples from our own previous study as well. So about 1,000 samples already collected for Gegc, and this number is rapidly growing as I speak. So these are some of the sites. We've recruited main sites in basically the whole of Australia, and we've also expanded that to the rest of the world. And of course, in India, we have LVP, Naraya and Netralala, and Aditya Jyot in Mumbai. But you can see this is a list of different places that are already being involved in this particular study, and the list is growing. So the outcome of this consortium is really to allow us to collect a large and standardized collection of patients in terms of data from around the world. It will increase our statistical power. It will allow different comparisons between ethnicities and geographic backgrounds. We'll be able to undertake clinical, genetic, and other risk factor information studies, such as looking at disease causation, progression, and response to treatment. And hopefully we can better define early detection of keratoconus and a universally accepted classification system. And also all of this data can be fed into AI applications in the future. So just to thank my co-executive members, Mark Daniel, Elsie Chan, and Srishna Sahab-Jada, and funding sources for this project. Thank you very much.
Okay. Thank you very much, Professor Baird. This is work that Dr. Natarajan mentioned in the previous session, and I also had the pleasure of learning more about it from your associate Srujna in Mumbai just a couple days ago. But this is any questions, any comments, anything that you would like, anyone? No? So Professor Baird is going to be here in other sessions as well as throughout. So please feel free, very approachable. And should we move to the next talk, you think? So that's basically me, and I've been introduced here several times, I guess. So, okay, that's fine.
So it's my great pleasure to introduce my co-moderator, John Prakash, from the NEI, and he's in the top floor of the NEI building, and I'm sure many of you are familiar with his name and what he does in terms of US-Indian collaborations, and also his prior involvement and ongoing involvement in GEGC. So, John, can we hear from you?
Thank you very much, Paul. Really appreciate the kind words. So I want to make another introduction. Dr. B.K. Jain, he's here. He's the founding chairman and of the Sadhguru Netra Chikitsalaya in Chitrakoot, one of the largest facility I have ever seen anywhere in the world in terms of service, cataract service facility, amazing, unique patient population. So if you're many of you were here in the previous session, and just to add to that if you're interested in looking at research collaboration, they're building a new research facility, and I'll have Dr. Alok Sen talk about it in a section. So let's get started with this. I'm also representing Dr. Santa Tumenia, who is the acting director and could not make it to this session. She sends her regrets and best wishes to all of you. So I'm going to take double time here. Okay, so opinions expressed here are my own. The U.S. government doesn't take responsibility. So, Global iGenetics Consortium. Many of you already know GEGC, and this was evolved. The idea was conceived in this part of the world. We were at the SARC meeting, Dr. Natarajan, myself, Dr. Takeshi Iwata, and in a interesting way that we looked at unique patient populations, so much enthusiasm, working with the SARC countries and especially folks from India. And this led to some discussion at National Eye Institute, and we assembled a group of pretty much all thought leaders, key leaders from around the world, and they gave us a recommendation that develop an international platform for exchanging information and data. We share genetic information to rapidly isolate common disease variants, establishing system for accurate diagnosis and disease subgrouping, establishing system for cost-effective genetic analysis, developing a big effort in going into genotype-phenotype database, which we talked so much about in the previous session, and supporting and fostering collaboration among the scientists interested in iGenetics, which is a big goal, which we very, very actively we have done, and collaborating with international and regional organizations. And it's just ideas are coming in. We'll be at the WOC in Cape Town. We'll be in Argentina at ICER meeting and several other. We always have a strong presence at ARVO. And many of you who are coming to ARVO, please remember that ARVO meeting, as you know, ends on Thursday, which is it will be from May 3rd to May 7th. But on May 8th, which is Friday, we are taking advantage of many of international visitors, dignitaries, and thought leaders coming to the Baltimore area, and the National Eye Institute NIH office campus is literally about 45 minutes, an hour from there. So we are having a symposium on May 8th. Please do let me know if you are coming to ARVO. I would like you to consider staying that night, Thursday night, and attending the NIH NEI, the symposium specifically on GEGC. And let me know. I'll send you official invitation, and you could be part of that. Okay? So this is my open invitation to you. Okay, so this is countries. We have presence all over the world, essentially all continents. There's a definite benefit to global participation. It creates a partnership, and we have access to big data. We all work together for the common cause. It leverages, we put in our own investment, but share our information for the benefit of humankind. We are interested in standardizing our data collection and all other particulars. It increases the benefit of our research dollars, as we talked about in the previous session, that NEI, its own budget is $825 million. NIH overall has $42 billion to invest every year. So essentially what NIH and NEI do, that sets a big direction into where eye research is going worldwide. So we have a very unique role to play, regardless. As you know, the money moves the research essentially in many respects, and a lot of new ideas come into us, and we take advantage of that, working with global participation. As I said earlier, that anyone and everyone can apply for NIH grants. It's not restricted to the only US people. And we have participations right now. I showed you the worldwide graph that people all over the world are working with us. So it actually gives a lot of patient participation and trials around the world. And we are standardizing the process. We are evaluating disease classifications and so on. So in many respects, we consider this as a translational effort which is leading into a global participation, and GEGC stands for all that. So, I'm not going to go through every slide here because some of this got covered in the previous session, and we, in addition to some big programs, like we consider India as a very active program, but if you look at our training and fellowship, and postdoctoral programs, short-term fellowship, now the new ICO fellowship, and the DBT collaboration and research, India is one of the most active program participant with NEI. China as well, we have programs working with Brazil, Ireland that are in existence. But I want to emphasize on these two fellowships that are highlighted. One is called NEI International Genomics Fellowship, and which is we select 5 fellows for 1 month. And this has been going on since 2016, and I'm delighted to mention and see that Dr. Shalja Tibrewal, who was the very first recipient of this worldwide, she is here. She's from India. Dr. Shalja, would you please identify yourself? And she is, and it's been a few years since she came back after the training, and she just last Friday, she organized a wonderful symposium at Shraf Hospital in Delhi, and made us all very proud, because that was the goal that we had set up 5-7 years ago. This is what we want to bring people, train them at NEI, send them back to their respective countries where they could start their own career, launch their own thing, and become thought leader one day. So, and Dr. Shalja has literally proven that into the best effort. We also have a new fellowship that we started, which is, it was just signed last Arvo, and the very first recipient, as he was pointed out, is Dr. Mayank Bansal, who is also here in this. So let's give a round of applause to Dr. Bansal and his worldwide selection. So you can see that India is not only excelling and also sending all these fellows and working with us, but we can see the benefits in future, because the two thought leaders are, the budding thought leaders are already here. So Mayank, we're going to put a lot of burden on you. And so when you come back, you'll be in a position to actually do lots of good things and work with us. So these are the links that we're establishing. So I would encourage many of you to really disseminate, and there was a discussion on that with Dr. Lalit Verma, Dr. Namrata, that how do we actually really advertise this in India and rest of the world. So we'll work on that. We're also starting new programs, and as GEGC is also expanding. So this is all effort for in the GEGC realm that, what we have been able to do and accomplish in such a very short period of time. So let's go to that. So we talked about this Genomics Fellowship. This is you apply in the month of, it starts in summer, essentially application, you get to know a few months later, and the program is a 1-month program, and we are doing it every year, and we are committed, very much committed. We had started out with 1, now we invite 5 fellows every year. Okay? And this is my contact information. So what I like to say is, good eye research anywhere is good eye research everywhere. And I have a patent on that, so that I truly believe in that. What I want to take advantage of is, since we have Dr. Santatuminia's time, I would like to invite Dr. Shalja Tibrewal here to talk about the symposium we just did last Friday at Shraf Eye Hospital. She's going to summarize that GEGC. That's a second symposium. And then, but I guess before we do that, we have Dr. Jan and Dr. Alok Sen here, and they are sitting in a very, very, hotbed of research in terms of unique patient population. They're also building a new research facility. So if either of you gentlemen like to say something, Dr. Alok, and then we have Dr. Shalja here.
Thank you. Thank you so much, Dr. Gyan Prakash, and good morning to you all. I come from a very small town in Uttar Pradesh, and the place is known as Chitrakoot.
Yeah, go up there, please.
So we have been working there for over 40 years now. There's a hospital named Sadguru Netra Chikitsalaya, and this is one of the largest eye care facility in rural India, if I can say so, sir. We are trying to set up, as the others have did in the past, like LVPI, Shankar Netralaya or Arvind. We are trying to set up a lab, research lab there at Chitrakoot. So we already have a very good training facility there, and we are recognized ICU center for training in different subspecialty in ophthalmology. I would like to speak on what difficulties which we are facing as far as setting up a lab, research lab is concerned. So attracting ophthalmologists to Chitrakoot is not very difficult because of the volumes of patients which we see there and the amount of surgeries which we do. But to get basic scientists and research fellows to Chitrakoot, a small town, is one thing which we face a lot. And we are still yet to find someone. So if you know someone, or if you are willing to come to Chitrakoot and help us in establishing a basic lab facility, we would be very grateful. We are already working with Shroffs and other centers in North India. We have formed a consortium, and already we have submitted a paper on coloboma-related disorders. We see a lot of these patients there. I think over last 10 years in retina department, we have collected around 1,200 patients of coloboma related eye disorders. So I think is the patient population there is quite different from what we see in other part of the country, and I look forward for more collaboration with other centers as we have made with Aditya Jyot and Shroff Eye Center in North India. Thank you.
Thank you very much, Dr. Alok. So what I wanted to basically highlight here, that there are opportunities and there are unique patient populations in India, and there are facilities and people who are willing, such as Dr. Alok and Dr. Jain here, to really work with you. So while you are here, please take advantage of the opportunity. So now Dr. Shalja, who will speak on the GGC specific symposium, that what we are accomplishing.
Yeah. So thank you, Dr. Gyan Prakash, for giving me this opportunity and actually introducing me to the world of genetics. So as clinicians, always the genetics chapter was always like something that we skip through and don't go through it because of all the high-end medical terms. But this fellowship which I went was really an eye-opener, and through that and coming back to India and working, I had my own struggles and working, and I realized that really we need to have we have the power of big data actually, but the problem is that in India actually to collaborate and to work together is a little bit difficult. Still these are a little bit of few of our attempts that we do, and the second annual GGC summit was something like that. We had done this summit last year and also around the same time around AIOS, and these are a few glimpses of the this year GGC meeting. It was on 7th of Feb. And Dr. Gyan Prakash and Dr. Paul Baird were there with us. And we had members of our Bodhya Eye Consortium, as Dr. Alok Sen mentioned. We are a group of hospitals from North and Central India, which we are forming kind of a consortium, dealing with our own problems together, collecting data, and forming our own databases, and eventually want to collaborate and work on genetic studies as well. So the meeting was attended by Dr. Paul Baird from Australia, Dr. Gyan Prakash from NEI, Shroff Charity Eye Hospital, Dr. Radhika Tandon from AIMS, it was represented by Dr. From Chitrakoot by Dr. Sood, and Sitapur, Dr. Madhu Baduria had come. We also had Dr. Mridul Sarkar from Islamia Institute. We have Arvind Eye Care System being represented by Dr. Sundaresan, and Aditya Jyot by Dr. Rajkumar. So it was a mix of scientists as well as clinicians. We had a discussion about what are the common and ocular genetic problems that we are facing. In fact, there's so many, but we could discuss a few of them at least in depth. And it was a kind of an...
Please come over here. So I think that the best, while she's settling down here, the best part was that in the two annual summits that we have done, the Indian team, GEGC team, we like to call it, have been able to pull together all the centers from around that have specific and unique patient populations, which is the accomplishment and which is one of the goals of GEGC. Okay, please.
Yeah. So we had people, like as I said, it was a good amalgamation of both scientists as well as clinicians, and discussing like our problems with the scientists, so that how could we approach it in terms of research. So as mentioned, we discussed a little bit about diabetic retinopathy, advanced retinoblastoma, Mack complex of disorders, congenital glaucoma and CHED. There's just to name a few. But the unique population in the sense that if we see any of these diseases and we pick up the literature, we have most of the genetic literature and mutations from the Western population. Now, if we want to replicate their studies and see it in our population, usually we do not land up with a significant result. I can give an example of coloboma, where worldwide we know that PAX6 mutations are most common, but there are studies from AIMS as well as down south from Arvind, where they have studied PAX mutations in our set of coloboma patients and not found it in a single patient among 30 cases and 30 controls. So our population is definitely different from the Western population. We need to explore, and there's where collaboration comes, because with the advent of whole exome sequencing and sequencing, we get a lot of variants which are of unknown significance. They are of unknown significance because they have not been described anywhere. But we have our own data. If we have that same VUS coming up in our set of patients, and then we can prove it as pathogenic mutation. So that's the advantage of having collaborations and having big data, especially for genetics, rather than everybody working in their silos. If you all collaborate and have a genetic database, it will be helpful. That's the way forward, I think, for the ocular genetics in India. That was the scope of this meeting. Anything else you want to add?
Yeah. So any question on Indian effort of GEGC? Yeah. Any question on what we have done so far in India is last 2 years we have brought the attempt is to bring the entire Indian contingent of GEGC together so that they can all speak with one voice. Last year we did part 1 and part 2 of the symposium. Part 1 was done at the Shroff Hospital under the leadership of Dr. Umang Mathur, and part 2 was done at the Aditya Jyoti Center in hospital there in Mumbai under the leadership of Dr. Natarajan and Dr. Radhika Krishnan. So this year we combined that all together and just did one, and people from all centers came. And so that's moving forward. They can all combine their effort and then speak with one voice. So that's the. I don't know, if Dr. Baird, do you have any comment on that? You were there too.
Yeah, no, I think it was one of those great meetings where there was a lot of ideas floated around, and you could clearly see the enthusiasm and need also as well to actually do something here and set up a whole series of different parameters, I guess, and different ways of collecting samples and talking to different groups. And I think we have a perfect example here of somebody who's come back from NEI who can do that.
Just giving an example that Dr. Alok mentioned, that we completed a paper on Mack complex of disorder. We just wanted to see how many patients do we see. It's not related to genetics as of now. We just collected the data, and over 1 year, we just had 3 hospitals combining, and we had 515 patients of Mack complex of disorders. So that's the power that India has, that we have so much data, but we have to tap it in the right manner to help our own patients. And the test panels which are available today, they are not probably perfect for the Indian population. We need to have our own data so that we can, help our patients in genetic testing as well. Most of the tests then go into dustbin only if we are not trying to correlate it with our patients. So that's the whole idea, ultimately.
So thank you very much, Dr. Shalja. So in India, if you essentially we are trying to put our efforts through Shroff Hospital under the leadership of Dr. Umang Mathur, Arun Arora, Shalja Tibriwal, and then Aditya Jyoti with Dr. Natarajan and Dr. Radhika Krishnan, Dr. Rajkumar. And we also, as you saw earlier, my co-chair moderator, Dr. Bhavna Chawla, So she's a professor at Ames, and actually she's here. I thought she left. She's already here in the audience. So Dr. Bhavna is spending some time with us at National Eye Institute, and she has been working with our leaders. So essentially, I expect that when Dr. Bhavna returns back to India later this year or in near future, she will also play a key role, an important role, in all India-wide in terms of iGene and GGC that she has worked with us and has been exposed to. So these essentially are the leaders of GGC in India, and they are connected with us on a very regular basis. So please communicate and channel your information. We want to see India as a one picture united. That's the goal. Thank you. So I guess our next speaker is.
Is Dr. Angul Wang here? Oh, yeah, good, okay. So Dr. Wang is going to talk to us about gene therapy for Leber's hereditary optic neuropathy.
Yeah.
Sorry, I don't have any other details from where you're from, so.
I'm also a member for GGC. And
Yes.
and no, not this one. I have already uploaded. That I'm the principal investigator for the reflex study of the GeneSight Biologic Company. We all know that lung disease is a maternally inherited genetic disease, and it was first described in 1871 by Dr. Theodore Leber, and it is characterized by acute or subacute bilateral visual loss, predominantly occurred in young men. And it is caused by a mitochondrial gene mutation, and we all know that there are three primary mutations, 3460, 11778, and 14484. Among these three primary mutations, 11778 mutation is the most predominant one. It occupies about 85 to 90% of the cases, and in Taiwan, it's almost around 86% of the case. And it will affect the complex 1 respiratory enzyme. We call that NADH dehydrogenase. And the 11778 will affect the subunit 4, so we call that ND4 fragment of the gene. And we all know that the visual outcome in the lung disease is very poor. In most cases, the vision will deteriorate to worse than 20/200, and there's also clinical evidence for further progression that continues long-term, year after years. And the spontaneous recovery rate is rather low, about 4% in the case of 11778 mutations. As for the treatment, there's a drug called idabenone, and which is a derivative of the coenzyme Q10, and which has been proven to be safe and well tolerated, and it can decrease the progression in the patient, and by a low-dose study in 2011. Another treatment option would be the gene therapy, and that's causing by deliver the synthetic ND4 subunit to reverse the mitochondrial deficiency of the 11778 mutations. And currently, the clinical trial is in the stage of 2-3 clinical trial. Around the world, there are three primary sites for the gene therapy for the lung disease. The first one on your left-hand side, that's the GeneSight Biologics. That's a French company that's based in Paris. It conducts multi-center studies, and the product name is GS10. And it has gone through the phase 1-2 study, which has been published. And the phase 2-3 study, it has two phase 2-3 study. One is called Rescue Reverse. The other one is called Reflect. And another center that was based in Florida, that's the Bascom Palmer Institute, that Professor John Guy lead this group, and they have been single center, still in the safety phase 1-2 studies. And another group that's led by the Professor Li Bing in the Tongji Hospital, that's in Wuhan. And that would be a single center studies too. And Professor Li told me that in their study is going to be going on to the phase 3 in this year, in the end of this year. We know that the GeneSight Biologic is a French-based company, and the product is originally called as GS10. Recently, they gave it a commercial name, which is called Lumivoc. And it's a product of recombinant AAV2 carrying ND4 fragments. And it has gone through the phase 1-2 study and also a phase 2-3 study that's called Rescue and Reverse. And the strategy they have been using is that the AAV2 will send the ND4 into the nucleus, where it is transcribed into the messenger RNA. And on this messenger RNA, it is tagged with a mitochondrial targeting sequence that's called the MTS sequence. This MTS will shuttle the messenger RNA of ND4 from the nucleus directly to the outer membrane of the mitochondria. And over the outer membrane of mitochondria, the ND4 protein are produced and then incorporated into the mitochondria to make it work. And this kind of strategy, we call that allotopic expression. Let's look at the safety of the GeneSight Biologic Company. And they have been recruited 15 patients and to receive a single dose of GS10 in the worst-seeing eyes. And these patients have an onset duration from 8 to 271 months. At about 2 years, they have been reported 96 treatment-emergent adverse events. 40 of them are systemic and 56 are ocular, and no serious adverse event relate to the treatment or the procedure. And no people die, and no treatment adverse event leading to the study discontinuations. Here's the table for the ocular side effect, and you can see most of them. They are, most common one would be the anterior chamber inflammation, vitritis, and ocular hypertension. All these kind of most commonly occurred adverse event can be managed with medications. And for the safety reports from the Bascom Palmer, they have been reported no one lost vision and no serious adverse event. Most of the adverse events are minor, such as a transient increase of IOP and exposure keratitis, subconjunctival hemorrhage, something like that. And the report from the Wuhan Tongji Hospital, they said that they have been recruiting 9 patients, and they report that no local or systemic adverse event relate to the vectors. So it looks like this gene therapy is quite safe for our patients. And let's look at how the clinical efficacy of these patients, of these drugs. And then in this, the first phase 2-3 study, they call rescue and the reverse. The rescue means that those patients have onset less than 6 months. And they have recruited 39 subjects, which they receive GS10 injection into one eye and placebo into another eye. For the reverse study, they recruit patients with onset between 6 to 12 months, and they have been recruiting 37 subjects. Also, they receive GS10 into one eye and placebo into another eye. There is another study conducted by the same company, which is called Reality. That's a retrospective and cross-sectional observational study for the Leber's disease to study the natural history of lung disease. They have been recruiting 15 patients. And they reported that in the April and the September press release from that company, they have reporting that there is a clinical meaningful improvement of 25 to 28 ETDRS letter equivalents versus nadir of best corrected visual acuity in the GS10 treated eyes at 96 weeks in rescue and the reverse study respectively. And there is bilateral eye improvement continues in the key visual functions such as best corrected visual acuity and contrast sensitivity. And its safety and the tolerability is well established over the course of the trial. And the recovery of the best corrected visual acuity greatly exceeds the results from the published natural history studies. Let's look at these figures. The black line is the data from the reality study. You can see that the vision will drop till come to a nadir at about 6 months to 1 year. Over there, that's we call the nadir. And then it will slowly progress. And for the red and blue line, they are the rescue and reverse data. The dotted line, it will be the rescue data. And the red dotted line will be the GS10 treated eye. And the blue circle dotted line will be the placebo group. And you can see that the vision drop and then coming back a little bit after the 1 year. And they reported that the rescue GS10 treated eye have a 25 letters better than the nadir conditions. For the reverse study, they are recruiting patients treated between the 6 months to 12 months. So they have been ejected around between the 6 to 12 months after the onset. So, and they are represented as the solid line, solid blue line and solid red line. And then you can see that the GS10 treated eye have a little bit better visual acuity than the blue line. And both eyes recover at the same time. And they have been said that the GS10 treated eye have a 28.1 letters versus improvement versus nadir. So the same company has tried to conduct another phase 2-3 study that's called REFLECT. The patient will receive bilateral IVI injection to mimic the real-world medicine. They have been open 13 sites, and it's planned to follow up for 2 years and recruit 19 patients. It has been started in 2018, and recruitment has been completed in 2019. There are 7 sites in the United States, 5 sites in Europe, and our hospital are honored to being selected as the only one site in Asia. In the informed consent agreement meeting, I will explain the informed consent in person, and there will be on-site translator and the translator grade witness and the IRB officer to watch over the IRB agreement. And for each follow-up visit, there will be an on-site translator that will be available for the international patients. And there, the informed consent has been translated into different language for each countries. And because the REFLECT data hasn't been analyzed yet, and so we only have the rescue and the reverse data. And in summary, clinical meaningful improvement of 25 to 28 ETDRS letters versus nadir of the best corrected visual acuity in GS10 single injection in rescue and reverse study was shown at 96 weeks. And the recovery of the vision greatly exceeds the results from the published natural history studies. And the REFLECT study will mimic the real-world outcome by using bilateral injection of the GS10. However, the data is still to be analyzed. And the last one is that according to their press, the GeneSight Biologic Pump Company will expect to share the data to the EMA and file for approval in Europe in the Q3 2020. Thank you for your attention.
Okay, this paper is open for discussion. Thank you, Dr. Wang.
Thank you.
Any comments? I just have one question for you. The data you've shown is obviously showing an improvement in rebound and rescue. So I was just wondering, you showed up to 100 weeks or around that.
96.
96. Do you have any data on sort of phase 1 studies going even further than that?
I think because the rescue and reverse was conducted by the company, and personally, I did not involve in that project. And according the 96 weeks data is the most updated one, just released from the December. And I think we don't have much more longer follow-up yet. But on the other hand, they also did another study that called a reality. That's retrospective and observational study that can observing the natural history of the lung disease. They can watch over the natural history of these patients. And so in that case, they have patients with a longer period of after onset to show their natural visual outcomes. And also you can compare from the data of the studies in the past, and they have a lot of data showing the natural history data without, at the stage of without treatment.
Right. Okay. Thank you. Inshallah.
I have one question, sir. In your slide where you have shown the data for the reverse and the rescue,
Yeah.
even the sham had improved.
Yes.
Yeah, so what is the difference between the sham and the treatment?
The difference between the sham and the experiment group is very minor, and that's the key problem for the company, because they have a lot of headache because you didn't show the clinical significance between the treatment and non-treatment group. And however, this phenomena is not specific for GeneSci Biologics. It's also reflected also in the data from Florida and also from the China, the Wuhan group. They also show the same bilateral improvement after a single injection into unilateral eyes. And later on, the GeneSci Biologics company has moved on to conduct an animal study. They inject the vector into the monkey eye, and recently they have a press release. They found out that once you inject into one eye, and after a period of time, you take out another eye and examine, you will find the vector existence in the other eye.
Essentially, even one eye treatment will help the other eye. I mean, injection.
Yeah,
not
and
required in other eye.
because in the prior time in the animal study before they launched the safety study, they have a systemic blood examination by PCR, and it was shown to be negative. However, if you examine the other eye, the PCR was positive. And so maybe transport through the neural system, like a chiasm or something.
Also, in our patients, we don't see that in natural history that all of them like reduce the way in the reflect study was there. In our patients, some patients do improve naturally also.
The natural recovery rate for the 11778 was quite low, but not impossible. Some of the cases were spontaneous recovery, but the recovery would be much lower than the 14484. That's around 4%. So some of the cases you will see some patients getting better.
Thank you.
Thank you.
Okay, thank you. Okay, the next speaker is Dr. Heather Mack from Australia, and she was, or is currently the first female president of RANSCO, the Royal Australian New Zealand College of Ophthalmologists, and she's going to continue on the theme of treatment, but this time looking at Lux Turner. So we'll see what's happening in that therapeutic space. Thanks, Heather.
Thank you, Paul. May I have my slides? Can you find my slides? Yeah, I uploaded them yesterday. Oh, what? Oh, this is good.
स्विच करना
That's it. Thank you, Paul, for that introduction, and thank you for the invitation to speak at your meeting. This is my disclosure. As you said, I'm speaking what you might call downstream from original genetic work, and I'm talking about implementing Luxturna from the perspective of a clinician and patients, and talking about these topics. So RPE65 gene, as we know, resides in the RPE and encodes a protein, RPE65. RPE65 is involved in the restoration of 11-cis retinol, so the vitamin A metabolism. And as we know, mutations in the RPE65 gene lead to degeneration of the photoreceptors in humans and in animal models. It's important to note that the phenotype of biallelic RPE65 mutation-associated retinopathy is variable, and that affects the implementation. So we see the phenotype, there's one occurring at congenital, which we call labocongenital amaurosis. It can be seen in childhood, which we call SICORD. It can be seen presenting in adults as a typical RP. Some of the patients have a classical fundus appearance, but many do not. With this heterogeneity, the only way to be certain that you're dealing with RPE65 mutation-associated retinopathy is to do confirmatory genetic testing. And when you do that, you find that about 10% of the cases of labocongenital amaurosis are due to RPE65. And in adult RP, about 2% of cases are due to RPE65 mutations. Everyone that works in RP knows that the mutation resides in the RPE, and secondarily we get dysfunction of the rods, and following that the cones, with this list of phenotypic features. In RPE65 mutation, because of the role of RPE65 protein, it's said that nyctalopia is more prominent than the other retinal dystrophies. Further to the point here that we have a lot of phenotypic variability, this is a study showing you the variability in decay of visual acuity and Goldman visual field over time. Now, we have, as we all know, we now have Retagene, Luxturna, as a licensed treatment for this condition. So the Retagene is a treatment which is a live, non-replicating AAV that has been genetically modified to express the human RPE65 gene. There are many other competitors in the space beyond the form of Retagene, which has been approved by the FDA. So all the other groups have had slight differences in the construct, the vector, the purification, etc. So we need to think about the competitors as biosimilars rather than actually identical to Retagene. Now, the phase 3 study of the Retagene has it being given as intravitreal therapy with a vitrectomy, subretinal injection, and conceptually the product, the AAV, is transduced into the RPE cells. Some of those have the cDNA, which is incorporated into the cells, and then we have expression of normal human RPE65 protein, which has potential to restore the visual cycle. Before we look at the pivotal phase 3 study, we need to consider how we assess the effect of this therapy and what are the outcome measures when you consider that the patients with RPE65 retinopathy all have low vision at the start. So visual acuity, etc., standard measures aren't very helpful. Instead, the studies have to look at the more sophisticated outcome measures such as mobility testing, full field light sensitivity threshold testing, and functional MRI. So for the FDA-approved study, which led to Luxturna, the investigators actually worked hand in hand with the FDA to design this MLMT study outcome, which is walking through a maze at varying light levels. So at baseline, all the subjects are unable to perform the maze at 1 lux, i.e. very dim circumstances. And the outcome is the improvement in the lowest light level in which they can navigate the maze, but not expressed in lux, expressed in a 6-point scale of improvement. So here are the results of the study here, the phase 3 study, which had 31 patients, 20 intervention and 9 control. Overall, 65% of the treated patients improved, but of course 66% had adverse ocular events. So if you look at the panel there with the lux score, and you can see improvement, it's not absolute lux as I said, improvement in their ability to navigate a maze in dim lighting, and a better improvement when both eyes are treated, they're the blue dots, and you can see the data out to 4 years. And similarly with the FST outcome, data out to 4 years, improvement, best improvement when both eyes are treated. You can see there the visual acuity outcome is a bit less convincing. Based on this published data, the FDA approved Luxturna, followed shortly thereafter by the European Medicines Agency, and the treatment is given to both eyes no less than 6 days apart by subretinal injection at a vitrectomy. Now, if we're looking at implementing, the durability of treatment is uncertain. The FDA-approved study showed data only out to 4 years. After that, unknown. We have longer data with the dogs, the canine model, so there's 5-year follow-up data for dogs, and interestingly, this study showed that at the treatment site, if there's 63% of photoreceptors present at the treatment site, there was a robust and long-lasting treatment response, whereas if the subretinal injection was given at areas with less than 63% photoreceptors, continuing degradation. I'm showing you here also some information from the other competitors of, as you will, biosimilars to Veretogene, not identical. But the study of Bainbridge there, which was looking at increased sensitivity on microperimetry, shows you there not all the patients responded at all. Some did, but those that responded had a decay by 3 years. And the lower panel there shows another competitor group, which when comparing control and treated areas, there was no effect on the ongoing retinal atrophy, though interestingly, even though the patients had ongoing retinal atrophy, a treatment response of increased retinal sensitivity was maintained. So I think all of this shows you that beyond 4 years, the result is uncertain. If we look at implementation of this data, there are obvious barriers. So obviously, this can only occur in specialised centres. No one has a maze in their office. The specialised centres need to have 12 mazes so they can randomly assign the patients. They need specialised pharmacy able to cover these very specialised genetically modified products, and registry for long-term follow-up. Then we have to identify the patients. Now, since it's ultra rare, the number of patients is very low. It's more or less like finding a needle in a haystack. It's said the phenotype has increased nyctalopia, but I'm yet to be convinced of that. And further, very few patients in the clinical setting have had genetic testing, and even those that have testing, it's not always certain that the mutation is pathogenic or biallelic, and certainly extensive genetic counselling is required. Beyond my scope here, obviously the best treatment window is in the younger individuals, but that's ethically problematic. The patients do not just stand up from the vitrectomy and walk away. They have to have extensive visual rehabilitation and support services. And the cost is very significant. In the USA, $850,000 for the 2-eye treatment, plus the cost of surgery, plus all the follow-up, for example, the cataract surgery they'll require after vitrectomy. Working out the cost-benefit analysis is very difficult. Here, I've put here one of the models. It obviously critically depends on the assumptions made about the duration of treatment. and it comes out to be very expensive. As you can see there, $645,000 per QALY. And this needs to be interpreted in the context of opportunity cost, what else you could buy with this money, for example, a Harvard education and a self-driving car. Now, the patient experience data is none in RPE65 gene therapy. There's a little data elsewhere in gene therapy trials, which is where we look at motivation, understanding and perspective. We find the patients expecting disease reversal, which is not the case. They have poor understanding of the process, but with self-interest, they're happy to spend taxpayer dollars. So Luxturna, a fabulously interesting treatment and a wonderful outcome of a whole lot of genetic research. Implementation problematic, watch this space. Thank you for listening to me, and please, you're welcome to World Ophthalmology Congress 2022, my hometown, Melbourne, Australia.
Thank you, Heather. Any questions for Dr. Mack? Yes, please.
I just have one comment. Very interesting talk, and I deal with inherited retinal diseases, so I deal with Leber congenital amaurosis as well in Florida. So I don't know. Talking to investigators later on, like the aftermarket analysis, how do you feel? What is the? I mean, objectively, we established that it was a treatment, but subjectively, how do the patients feel and how do the patients' parents feel, whether there was any improvement? I heard about your comment on the night vision, but the mobility as well.
Yeah, there are many unknowns there. I've not found published patient clinical data or patient experience data, and for me, that's the important area of follow-up, because we're trying to identify these patients out there in the wild, so to speak. Trying to identify them, we need to be looking at their patient experience, patient understanding, and then on the tiny minority who are treated, we really need to follow them up very closely to see that there's value for money. They might be better with a self-driving car and an MD from Harvard.
Correct. Well, I like to be optimistic, and I think one thing that has happened is with that price tag for the Leber congenital amaurosis treatment, there are more and more pharmaceutical companies that have suddenly become interested in inherited retinal diseases, and so that's a great thing that has happened.
Yeah, it's fantastic. Watch this space.
I think what this study shows is that certainly if you can identify mutations in genes, certainly monogenic diseases, then there is a potential to actually develop therapies. So I think we've seen the first wave of therapies now that are coming out, and I'm sure there'll be many, many more. But whether they all have the same price tag, I don't know. But I mean, $850,000 for a bilateral disease, well, it's a lot of money. But you have to weigh that up against, will it improve somebody's vision, sight, and retain it for X number of years? So this is a question for governments, health regulators, but I don't know whether it would be funded at that level in India, because it would use up a lot of your health budget, I would imagine.
Even in USA now, they are talking to the government about what should be the final price, like Medicaid. It's not going to be that price.
And I mean, in the UK, the NIBSC has done a special deal on this as well with treatment to try and lower the price. So very interesting how the dynamics work, but obviously the company needs to get its product to the market, but it has to be at a fair price, which the government can afford or stand.
Interesting comments. I think the cost-effective studies should be part of, and they are increasingly becoming. Health economics is becoming a big issue. Outcomes research has definitely been part of our fabric in the US at least, and it's becoming in India as well. And see, this is where I always say that with the Indo-US collaboration, we can learn from each other. There are a lot of very cost-effective treatment, and especially with mobile devices, things are happening here at LV Prasad and Shankar Nethralaya and many places. So it's not only that, the India learns from the US and Australia and, other countries, but we also learn from India and countries in this region. But I want to thank all the speakers. Dr. Raj Rameshwar from South Africa, he could not be here, unfortunately. So one of the things that what we do, Dr. Takeshi Iwata, who is current president of Global Eye Genetics Consortium, he always reminds me in the morning before that, please make sure that the entire audience as well as the people sitting at the podium can come in front and we get a group picture. So please join me here for the group picture. All of you, everyone, just come and please line up here. And I'm going to give my camera, and official photographer will also help us. And that will basically conclude this session. Some of us are going to be in the background over there, so please bring your questions. We are very willing, and especially Dr. Paul Baird, who's the current vice president, and he'll tell you all about GGC. So please come in front, all of you. I think you can stand up here. Please come. Yeah.
Okay, everybody, quickly do this to quickly go.
Please come, please.
Oh, yes. Nice to meet you.
Please come. Dr. Chokshi, please come. Paul, please come to me. You're the official office bearer. Please come in. Please join.
Oh, quick.